Today we're talking about renal physiology, chapter 25, and we noted there are three steps in the process of forming urine: filtration, reabsorption, and secretion. We talked about glomerular filtration and the regulation of the glomerular filtration rate in the renal corpuscle, and then we started the discussion of reabsorption of that fluid in the tubule. We know we have to reabsorb most of that fluid in the solutes as the urine is formed, or we would lose volume very quickly. So the next step after the proximal tubule is the distal or the loop of Henle, We're now going to talk about the reabsorption of fluid in the loop of Henle of juxtamedullary nephrons. We're going to see how the fluid is reabsorbed and what the consequences of that are. So let's just take a look. The loop of Henle of juxtamedullary nephrons captures the fluid from the proximal tubule, and it's made up of simple squamous epithelial cells that are permeable to water but not to salt. So in the descending limb, we reabsorb some water from the filtrate, and then as the filtrate begins to ascend back towards the cortex of the kidney, the thin ascending limb, the simple squamous cells are permeable to salt and not to water, and we reabsorb some salt from the tubule. But then the thick ascending limb of the loop of Henle is made up of chunky, transport powerful cells that have a lot of mitochondria and a lot of primary active transporters. We're going to pump salt out of the filtrate into the interstitial fluid where it will accumulate, producing a very salty medullary interstitium. So this diagram will kind of illustrate what I'm talking about. You can see just a bit of the most lower parts of the proximal tubule there, but the thin descending limb of the loop of Henle allows us to reabsorb some water out of the tubule because of the properties of the cells. They're not permeable to salt. In the ascending limb of the loop of Henle, those simple squamous cells are permeable to salt and not water, and salt begins to diffuse out of the tubule into the interstitium. But in the thick ascending limb of the loop of Henle, we see all these primary active transporters that are going to pump, using ATP, pump sodium chloride out of the filtrate into the interstitial fluid, where it will accumulate, and I just drew a lot of sodium chlorides out there to illustrate the fact there's a lot of solute out there. There's a big concentration difference between the filtrate now and the fluid around it in the medulla of the kidneys. So together, the different parts of the loop of Henle produce a very salty medullary interstitium. Another thing we should pay attention to is what is the composition, or what is the osmolarity, the concentration of the filtrate at different parts of the loop of Henle, and consequently the collecting duct where it will then travel. So I'm going to skip to this picture, which is just a graphic of what's stated in that slide. As the filtrate travels down through the descending limb of the loop of Henle, we said that water will be reabsorbed by osmosis in that descending limb, and the filtrate will therefore become more and more concentrated. In fact, it will match the concentration of the medullary interstitium, and it may reach osmolarity as high as 1,200 milliosmoles per liter. And then the fluid begins to ascend in the thin ascending limb, and salt begins to diffuse out of the tubule, making the fluid more concentrated as it goes, and eventually we reach the thick ascending limb of the loop of Henle, and we just actively pump salt out of that filtrate, and what happens? It becomes more and more dilute. Medullary interstitium becomes concentrated, but the filtrate becomes very dilute. In fact, it's much more dilute than plasma, and that's really important. It may become as dilute as 100 milliosmol, or maybe even 50 milliosmoles per liter, as compared to 300 in the plasma and in the filtrate formed in the glomerulus. So some very dilute fluid is going to enter into the distal convoluted tubule. So that's the function of the loop of Henle in the process of forming urine. All right, let's take a look at reabsorption in the distal tubule. Reabsorption in the distal tubule is like a fine-tuning region. We have several hormones that will play a role in speeding up or slowing down reabsorption of salt and water and various constituents in the distal tubule. These are things that are familiar from our discussion of chapter 16, the endocrine system, chapter 19, the cardiovascular system, where we were very concerned with the volume of blood and therefore its effect on blood pressure. Let's just review those hormones that control functions of the distal tubule. First of all, aldosterone. Aldosterone is released from the kidneys when the blood pressure goes down. If those afferent arterioles, and therefore the juxtaglomerular cells in the kidneys, sense a drop in blood pressure, they'll release renin, and renin is an enzyme that will convert angiotensinogen, a plasma protein, into angiotensin 1, and angiotensin 1 is spontaneously converted to angiotensin 2 by endothelial cells, enzymes in their membrane, in the lungs predominantly. So as angiotensin 1 circulates through the body, it's converted to angiotensin 2. Angiotensin 2 is a powerful vasoconstrictor, as you know, but it also triggers the release of aldosterone from the adrenal cortex. So now we have aldosterone coming out, and aldosterone will cause the distal tubule cells to place more primary active transporters in their membrane, reabsorb more salt. And as we pump salt out of the filtrate, water is going to essentially be dragged along with it by osmosis. The fluid's getting more dilute in the tubule, more concentrated surrounding it, and that creates a gradient for osmosis. So we reabsorb salt and water. That allows us to retain fluid or increase the blood volume, assuming that we're drinking water. We're not making water here. If you continue to drink water at a normal rate and we reabsorb more water, the blood volume will go up, blood pressure will go back up. It also so happens that this is a hypertonic solution, so we're going to raise the concentration of sodium in the body fluids, and that's one of the important functions of aldosterone, retaining salt, maintaining the salt concentration as well as the fluid volumes, and we're going to secrete potassium. We'll come back to the subject of tubular secretion in a little bit, and we'll reiterate that potassium is secreted in the distal tubule, meaning added to the filtrate during the process of active transport under the control of aldosterone. A&P, atrial natriuretic peptide, is produced by the atrial endocrine cells. There's some hormone-producing cells in the atria of the heart, and when the volume of blood begins to rise, we said that if blood volume increases, venous return increases, that stretches the atria. We mostly considered how it stretches the ventricles and what happens with respect to the force of contraction. Well, now we're talking about the atria. It'll stretch the walls of the atria, which will cause atrial natriuretic peptide to be released so that the volume doesn't continue to rise catastrophically. So A&P, atrial natriuretic peptide, will be released, and it will go to the distal tubule cells and inhibit salt reabsorption and water reabsorption by osmosis. It'll also inhibit the production of aldosterone, the release of aldosterone. So A&P is like a check system to avoid runaway increases in blood volume and blood pressure. Once the blood volume begins to rise, A&P will start to block that process and, in fact, inhibit salt and water reabsorption. Okay, parathyroid hormone. When blood calcium goes down, you may remember from talking way back in chapter 8, I believe, no, chapter 6, oh wow, all the way back to chapter 6, talking about bones. And when blood calcium goes down, the parathyroid glands on the posterior of the thyroid glands releases parathyroid hormone, and they are the receptor for that negative feedback loop. They release parathyroid hormone, which causes release of calcium from the matrix and bones by the activation of osteoclasts, production of vitamin D, so we can reabsorb calcium from the diet in the small intestine, but also, now we're taking a look at the distal tubule. It increases calcium reabsorption in the distal tubule. We're not going to pee out calcium when the blood calcium is going down. We're going to retain that calcium, reabsorb more of it. And finally, antidiuretic hormone. ADH is released from the posterior pituitary gland, essentially the hypothalamus, when blood pressure goes down. Also, if blood osmolarity goes up, if the water is being lost from the blood, say through sweating, or expiring water in the expiration of air, evaporation from the surface of the skin, if we're not taking in enough water, we start getting dehydrated, and the osmolarity rises, the blood pressure goes down, and ADH will be released. ADH is going to cause reabsorption of water in the collecting ducts. So that's the last step of reabsorption, because after the collecting ducts, we're actually talking about urine that's released into the calyces and then into the ureters. So that last step is reabsorption of pure water under the control of ADH. What is going on there? Well, the collecting ducts are made up of epithelial cells that are impermeable to water, so that the filtrate would go right through them like a plastic pipe unless there's ADH present. In the presence of ADH, those cells place water pores called aquaporins in their membrane, and then water can be reabsorbed in that as the filtrate travels through that salty medulla. Water can be reabsorbed from the collecting ducts by osmosis. There's just a concentration difference between the filtrate, very dilute, and the interstitial fluid, very concentrated, and we reabsorb water by osmosis. Without ADH, we won't reabsorb any water in the collecting ducts because they don't have any aquaporins in their membrane, and consequently they're essentially waterproof. So here's a little summary slide of reabsorption in different parts of the nephron. There's also some mention of secretion here and there, and we haven't really talked a ton about secretion yet, but in the proximal tubule, we said that 2/3 of the solute in water filtered at the glomerulus are reabsorbed. And there that box describes that process. 65% of all that volume and all the salts are going to be reabsorbed. All of the glucose, all of the amino acids, all the important organic nutrients are reabsorbed in the proximal tubule, the heavy lifting zone of tubular reabsorption. So happens that hydrogen ions are secreted there, and some drugs. We said that there's secretion of some things. Well, there's an example. We'll talk more about that later on. Okay, the loop of Henle. Descending limb, we reabsorb some water. Ascending limb, we reabsorb some salt by passive diffusion in the thin ascending limb, should have been written in there. But then we reabsorb a lot of salt by primary active transport in the thick ascending limb of the loop of Henle. So a lot of salt is now accumulating in the interstitium, and that kind of increasing maroon color is sort of reminiscent of an increasing salt concentration as we travel deeper and deeper down into the medulla. So up here, of course, is the cortex of the kidney, and down, starting right here and down, that's the medulla, that's the renal pyramid, and things get real salty down in there in that interstitial fluid that's surrounding the tubules, surrounding the loop of Henle and the collecting ducts. All right, the distal tubule, reabsorbing salt and water by osmosis under the action of aldosterone, under the control of aldosterone, reabsorbing calcium under the control, fine-tuning by parathyroid hormone, secreting some potassium. We have talked about that a lot. We have to add, secretion means to add something to the filtrate from the blood after the glomerular filtration step. So we're secreting potassium. We've got to keep the blood potassium levels constant. And so we're going to, whenever you eat potassium in your diet, it all will be secreted in the distal tubule. And then finally, the filtrate travels down through the collecting duct and will reabsorb water under the control of ADH. How much water? We have to talk about that. ADH causes the collecting duct to become permeable to water, and water is reabsorbed by osmosis in that process. It turns out that actually urea, another waste product, an amine, a nitrogen-containing waste product, is reabsorbed in the collecting duct, and it accumulates in the interstitial fluid along with the salt. So part of the salty medullary interstitium is actually urea, as well as sodium chloride that's out there. Atrial natriuretic peptide, as we just said, blocks reabsorption of salt and water in the distal tubule. I drew this little line probably in the wrong place. I should have drawn it up here to block that reabsorption of salt and water. When the blood volume begins to rise, A&P is like a check on the aldosterone system of increasing blood volume. All right, so next time we'll talk about tubular secretion, and we'll talk also a little bit about how the kidneys can concentrate and dilute urine to accommodate the needs of the body.